Our last speaker today is Caren Helbing, and Caren is a full professor at the University of Victoria, and she's the recipient of the 2025 ICEDA Career Award. Caren is very well known internationally for her work on endocrine disruption, particularly on thyroid hormone signaling, and using those often with amphibian models. And so she's going to give us a talk today, as listed on this slide, Insights from the Frog Lady, and then building an understanding of thyroid hormone detection, signaling, and disruption. Caren, looking forward to your presentation.
Thank you, Glenn. First of all, I'd like to thank my colleagues and the selection committee for this honor, and I'd also like to congratulate the other three awardees for this year. And I acknowledge that I live, work, and play on the territories of the Wasanich, Lekwungen-speaking peoples, Kasepson, and Songhees nations, whose relationships to the land continue to this day. Thyroid hormones. We've heard about them already. They're super important in growth and development and metabolism of all vertebrates. There are two major thyroid hormones. There's thyroxine and there's triiodothyronine, T3. T3 is considered the more bioactive form of the two, but all of these hormones are the same in all vertebrates. One of the most striking examples of the importance of thyroid hormones is their role in amphibian metamorphosis. Yay, frogs. Thyroid hormones are the trigger for transforming a tadpole into a frog. Premetamorphic tadpoles are functionally athyroid. They have a thyroid gland, but you can induce a precocious metamorphosis simply by injecting the pre-metamorphic tadpoles with thyroid hormone if they're big, like the bullfrogs tadpoles, or if they're smaller, like most tadpoles, you can just immerse them in water containing thyroid hormone. Now, as an undergrad, I thought, I found out about this, and I thought, cool, I want to study this and find out how it works. So I did my PhD at Western University in London, Ontario with Burr Atkinson. And this is me in the lab, and it's really messy and stuff like that. My lab is much cleaner than this nowadays. I studied thyroid hormone-induced changes in gene expression in the bullfrog tadpole liver. That was my PhD thesis. That was in the days when cloning a gene was a big deal and cloning through the internet did not exist. Now, this is a figure from my thesis showing differential RNA levels using northern blots, for those of you who know what those are, using Set to make my figure and onion paper, for those of you who may remember, before digital photography. And a more modern version over here shows qPCR data of thyroid hormone receptor encoding transcripts in the tail fin using qPCR. Now, the northern blot here looks at liver expression of transcripts, control versus thyroid hormone treated. And in both cases, they show the sensitivity of tadpole tissues to thyroid hormones. Now, during my graduate work, I learned a lot of cutting-edge molecular techniques at the time, including these northern blots, southern blots, western blots. There's no such thing as eastern blots as far as I know, but I probably would have learned that too. PCR, protein labeling, and 2D gel electrophoresis of proteins, which is the older version of proteomics. Haha. When I started my independent research career, identification of toxic chemicals in water was limited to dead fish and the concept that the dose makes the poison, but not with hormones. It was becoming clear that through the work of many people, including Theo Colburn, John McLaughlin, Pete Myers, Lou Gillette, Tom Zoller, and others, that low-dose chemical exposures could alter development, causing deleterious sublethal effects impacting later life. So the timing of the exposure, especially during early development, can be more important than the dose. A defining moment for me professionally was being offered a chance to speak at the Environmental Endocrine Disruptors Gordon Research Conference 26 years ago. It was here that I met key players in the field, including the people I just mentioned, and many of them would become my mentors. There was a noticeable gap as far as understanding and detecting thyroid hormone disruption. So I took up the challenge to help close that gap with my molecular experience and experience with fish and wildlife. So my team at the University of Victoria and I focused our attention on creating and using various omics approaches, including transcriptomics, proteomics, and metabolomics that could be applied across animal species. We needed to define what's normal to help us identify when disruption occurs in the quest for bioindicators of thyroid hormone action. It wasn't easy, as we were often working with species that weren't considered models yet, like the bullfrog tadpole, for example. We studied a whole bunch of different chemicals, from pollutants, personal care products, pharmaceuticals over the years, including, I think, all the ones that were mentioned today so far, and using tadpole metamorphosis as our indicator and developing tools to monitor gene expression at the molecular level. Now, a notable example of this is triclosan, which is widely used in personal care products, including soaps, toothpaste, and disinfectants. There were concerns about its environmental persistence. They knew that it was toxic at high doses, and there were some inklings about potential endocrine disrupting effects. But interestingly enough, thyroid hormone wasn't part of that picture up until we started doing some work on that. But based on the chemical structure, you can see here, triclosan looks an awful lot like thyroid hormone over here. So what we did was we took pre-metamorphic tadpoles and immersed them in different amounts of environmentally relevant concentrations of triclosan for 4 days. And then half of the animals were injected with a dose of thyroid hormone, indicated here by the arrows, that would induce a precocious metamorphosis. The tadpoles were continuously immersed in these different amounts of triclosan as moving forward. And what you can see is that while triclosan had no effect on hind limb development here on its own, that after thyroid hormone injection, there was a huge acceleration compared to just injected animals alone with thyroid hormone. Those that were exposed to triclosan actually showed a marked acceleration in hind limb development. Now, again, these were at environmentally relevant concentrations. Interestingly enough, 2 days after the T3 injection, we were able to detect tissue-specific molecular changes well before noticing the hind limb effects, which were evident at 11 days. And here's an example from tail fin. We also looked at brain and so on. And you can see here that this is qPCR data that in that thyroid hormone itself, compared to the control in white here, induces the beta thyroid hormone receptor gene expression. This is at transcript levels, and that any dose of triclosan actually attenuates that induction. We wanted to see whether another frog species, Pseudacris regilla, which is a Pacific tree frog, and they're super cute, were sensitive to triclosan too, and indeed they were. Now, we teamed up with Vicki Marlett, who's was asking some lovely questions today, and we examined the environmentally relevant concentrations again of triclosan, both in the presence and absence of thyroid hormone. This time we used immersion of the tadpoles shown here in thyroxine. And because the tadpoles are smaller, it's hard to inject them. And then did a similar experiment with the triclosan immersion as well. So what we found is that triclosan resulted in kind of a complicated thing. So in the absence of thyroxine, there was indications of a stunting or a slowing down of hind limb development at the highest environmentally relevant dose that we tested of triclosan. In the presence of T4, it's the opposite. There's an acceleration of the induced T4-induced hind limb development. So looking at other features and so on, including molecular ones, the treatment of triclosan resulted in uncoordinated development with and without thyroid hormone. And here are some data that again, are qPCR data looking at the expression of the alpha and beta thyroid hormone receptors, and then some other thyroid hormone responsive gene transcripts. And all the red stars that you see there are indicating significant differences either with or without thyroid hormone present. Again, consistent with this uncoordinated development kind of scenario. Now, this work spurred on other work in other organisms, including rats and so on. And they also found thyroid hormone disruption at these low environmentally relevant doses. And the result ultimately was that the Canadian government has now listed triclosan as toxic for the environment, including endocrine disruptive effects. Now, while qPCR is a great workhorse for a small number of key bioindicator transcripts, we created unique microarrays with hundreds of gene probes that were designed to work on both ranids, including bullfrogs, and xenopus species. And with these in collaborations with researchers at the US EPA, we were able to uncover gene expression signatures in tissues such as the xenopus brain, shown here, upon exposure to known thyroid hormone disruptors like perchlorate, methimazole, propylthiouracil, and comparing that to thyroxine over here. And then we soon began experimenting with RNA-seq approaches and realized that it would be so nice to have a reference genome for American bullfrog in order to do this effectively. So over several years, we teamed up with the Nunch Barol's group at BC Cancer and came up with a first ranid genome or TrueFog genome assembly in 2017. Now, that wasn't a small feat, as the genome was twice the size of the human genome, and it was difficult to put it together. We now have a chromosome level assembly that'll be released soon of a male that will complement a recently released assembly of a female from Chinese researchers. So stay tuned for that. Using the genomics resources that we built, we performed RNA-seq experiments to compare tissue responses at 48 hours post-injection of thyroid hormones. I'm showing you data here from injection with T3s, 10 nanomolar T3, into bullfrog tadpoles. The number and proportion of differentially expressed gene transcripts are significantly higher in the olfactory epithelium compared to all the other tissues shown here. There's a whopping 38,000 contigs that are differentially expressed. We also see interesting profiles when comparing T3 induction with comparable T4 amounts. And while there are a number of contigs that do overlap between the two, there are many that are unique to each, which indicates separate signaling pathways. And there's also very little overlap that's observed when tadpoles are exposed to equivalent amounts of estradiol, where there's a tiny little dot right there. You can barely see it. For the olfactory epithelium, gene ontology or GO enrichment analysis revealed 5 clusters of enriched GO terms that are implicated in RNA regulation following either T3 or T4 treatment. And these clusters include spliceosome activity, microRNA mediated gene silencing, RNA polymerase activity, nuclear export, and RNA methylation. This suggests that the tadpole's ability to sense its environment through olfaction, like sensing predators and food, may be compromised if certain aspects of thyroid hormone signaling are perturbed or activated at the wrong times. The olfactory system remodels extensively during metamorphosis with close links to behavioral responses important for survival. So there's a marked shift from a plant-eating, aquatic prey item, the tadpole, to a carnivorous hunter, the frog, that includes a massive remodeling of the olfactory system. We know that the olfactory epithelium is key to sensing info molecules through testing for avoidance cues. This is an eye maze, and we can get an avoidance response from a tadpole with an amino acid mix by applying the olfactory cue on one side and a blank on the other side simultaneously, and then watching the tadpole swim over to one or the other area of this eye maze after this chamber is lifted up. So here we can show a typical response where the time spent that the tadpole was in the eye maze on the blank side was greater, significantly greater than in the avoidance cue side of the eye maze. Interestingly, if the tadpoles are treated with T3, this avoidance cue is gone. Even more interestingly, or additionally interesting, is that if we do T4 treatment or an estradiol treatment, there's no effect. So it's like the control. We also did these kinds of experiments looking at different kinds of effluents from municipal wastewater treatment facilities and stuff like that. And I'm just showing one result here. This is showing that an avoidance response can be altered by municipal effluent that has a defined pharmaceutical and personal care product cocktail, which was comprised of 15 chemicals that are commonly found in municipal wastewater at those concentrations. And this cocktail was added as feeder stock prior to actually looking at treating the tadpoles with the resultant effluent. And you can see here that this cocktail effluent also abolishes the avoidance response. So collectively, these results show that an effective behavior outcome can be measured here that is directly important for survival, that can be used as part of defining an adverse outcome pathway. Now, for the last bit of my talk, I'm going to switch gears to talk a bit about some of the metabolomics work that we're doing, which is really cool. And here we're showing some QTOF tandem mass spec results looking at serum metabolites during bullfrog metamorphosis. And you can see like going from A to G, pre-metamorphic tadpole through metamorphosis to the froglet, that you can distinctly define based on metabolomic profile these stages. And we identified several metabolite features that were individually distinct within those phases. But rather than go into the details about that right now, which it's really cool, but I'm going to show you something that's even cooler. And that is, it's hot off the press work that was done by very talented postdoc Lika Poulsen who joined my lab a couple of years ago and now has secured a faculty position at Copenhagen University. And she, along with Helena Petrosova, who's a senior research associate at UVic Genome BC Proteomics Center, set out to do something that hasn't been done before to the scale, which is applying mass spectrometry imaging, or MSI, to identify metabolites of the brain during frog metamorphosis. Now, MSI, what it does is it combines the spatial information of histology with the power of mass spec to identify where metabolites are located within a sample. So Rikke and Helena were interested in the lipidome, and ultimately we're looking for lipid biomarkers of brain development. So the interest was looking for indications of neurotoxicity. Now, this isn't a small feat, as it's technically very challenging, and typically it's been used to look at like solid tumors and mouse kidneys and brains. Rikke and Helena were looking at mushy tadpole heads and a bit firmer froglet heads, and wanted to directly compare them. So the first step was to properly generate cryosections of the heads at comparable planes. Then what they do is they spray a matrix onto the slide to help the ions fly when zapped with a laser, shown here. This is what a MALDI timsTOF Flex looks like. This is the machine that does all the magic. It collects ions that are detected by a mass analyzer here, and a mass spectrum is collected at each 50 micron pixel of the histological section. And these mass spectra are then used to construct images of the slides based upon a single mass to charge feature at a time. And in that way, you can actually look at thousands of metabolic features captured from that one slide. This is what they ended up with after a lot of troubleshooting and cleverness and methods development. Here we've got 3 tadpole heads shown here. This is a coronal section. You can see the eyeballs here in the brain. And what you're looking at is kind of like a heat map where yellow shows higher amounts of a particular metabolic feature and blue shows lower amounts of that feature. And this just happens to be a lipid that just beautifully highlights the overall head in a very, very beautiful way. So here's a representative metamorphosis series with a pre-metamorphic tadpole on the left, metamorphic climax in the middle, and then we've got the froglet on the right. And we put them all on the same slide, so we would not have to deal with matrix effects. So they're directly comparable to each other. And you can clearly see lipid features associated with the brain regions. Here's a phosphoinositol, cardiolipin, and then diacylglycerol. Examining the brain and brain homogenates, there's a significant increase in diacylglycerol shown here. This is the brain homogenate data, and here you can see the localization in the histological sections. And also, I want to mention that this is proof that frogs can actually smile at you. We then wanted to apply MSI to bullfrog tadpoles exposed to a known endocrine disruptor, PFAS, which we've already heard about through Lai's talk. Thank you, Lai. And what we did was we used 5 environmentally relevant PFAS doses, and basically the experimental test setup resembles a 48-hour pulse exposure that could happen in a pond. And pre-metamorphic tadpoles were primed with thyroxine under controlled conditions, and then the heads were collected for MSI, serum for lipidomics, thyroid hormone, and PFAS quantification, and then brains were also collected after MSI for further lipidomics work. So we were able to measure PFAS in the samples using an established MS method, and not surprisingly, PFAS levels went up as the doses went up in both the serum and the brain. And then we found evidence of lipid feature perturbation in PFAS-exposed tadpoles. I'm just highlighting one here, and that is the diacylglycerol that I just told you about during natural metamorphosis. It turns out PFAS exposure actually decreases the level of diacylglycerol in the brain region. You can see the control right here, and you can see at a low dose of PFAS that brain region is cleared. Diacylglycerol is an important second messenger, a signaling molecule in brain development, and it regulates protein kinase C, prostaglandins, and so on. And dysregulation of diacylglycerol can actually have a deleterious effect on brain development. Now, what really surprised us is what happened to PFAS itself. It was hugely concentrated in the olfactory epithelium, which is really strange. Here we go. It's glowing. And the other thing is that most of the brain is actually missing PFAS exposure here. What's pointing at right here is the pineal gland. So the signals that we actually saw in the brain homogenates are coming from the pineal gland. Now, we're currently examining the impact of PFAS, looking at the different lipid profiles and so on. We're going to be following up experiments, looking at the IMASE experiments and additional molecular analyses. So stay tuned for that cool stuff coming. So today's take-home messages are: we've developed several novel ways to look at thyroid hormone disruption using frog tadpoles that are super sensitive to thyroid hormones. We've developed RNA-seq techniques and found, well, that many tissues are responsive to thyroid hormone. The olfactory epithelium is extremely sensitive. And we also have shown that there's a link between thyroid hormone signaling through the olfactory epithelium and behavioral responses. We've developed a method for generating beautiful MSI images from very difficult samples. And those data generated give us new hypotheses for deleterious effects of PFAS, like disruption of olfaction, which was surprising to us. And we've also detected disruption of components that of the lipidome that could potentially act as biomarkers for thyroid hormone disruption and neurotoxicity, and provide further insights into PFAS's effects. And this work could not have been done without the large number of talented people that were actually doing the work. Here's a photo of recent members of the lab. I thank you for your attention, and I'm happy to take any questions. Thank you.
Thanks so much, Caren. I really enjoyed your presentation, and I was a little bit scared by some of the movement down history, and I do remember using Letraset, and
I thought you would.
when northern blots were all the rage.
Yeah.
But Romy, are there any questions in the chat?
We haven't question yet.
Then I'll have a question. I was intrigued. I was intrigued by the link to the olfactory epithelium. I mean, my recollection was that when we think of thyroid dysfunction in humans, we don't really think of there being an olfactory deficit. And do you think that this tie-in between, the olfactory target in amphibians is due to the transition from the aquatic environment to the terrestrial environment?
Well, it could very well be that. I mean, certainly it's a very marked change. Like the olfactory system, like completely remodels itself and it's very, very thyroid hormone dependent and sensitive. With respect to comparing it to humans, well, a similar kind of transition, it happens around birth. If you think about, a baby getting born from an aquatic environment and all of a sudden, voila, life on land, kind of thing. There are many, many parallels that happen physiologically and at the molecular level, developmentally, that have to happen in order for the newborn to be healthy.
Sure.
And so I would argue that the role of thyroid hormone in the case of olfaction has literally been just not on. It hasn't been on people's radar at all.
Yeah.
So it's very intriguing, and it's a great question, Glenn.
Well, thanks. That was very, very interesting. Any questions come in yet, Romy?
We have a question from Vicky. Dr. Caren, sorry. With some of these new lipids, thyroid hormones and olfactory epithelium, thyroid hormone potential mediation, and other discoveries suggest we are missing some key adverse effects in OECD USEPA standardized tests or our Canadian chemical screening regimes. Any new implication for risk assessment based on your research?
Oh gosh, you're going for the jugular, aren't you, Vicky? Yeah, definitely. There's no question that there are potentially some key. I'm hedging a little. Potentially, there are things that are missing that are quite important in the OECD guidelines and so on. And yeah, all we can do is provide the science and do a good job there and work towards improving those guidelines.
It's interesting. I'm not sure the history, if we go back, but it was probably in about the 1920s that people started to study the involvement of thyroid hormones in
Late
metamorphosis.
1800s.
The late 1800s.
Yep.
And it's just very interesting how over the course of that time we keep identifying new and different targets for thyroid hormone action. Yet alone, we're learning about how other chemicals in the environment can, interfere with those processes. It's just something that, I guess you never know everything, but you'd think that metamorphosis would have been one of those topics where we would have we wouldn't be making as many fundamental discoveries that we still are making.
Yeah, it's an interesting commentary, about how science goes. And so some key discoveries were made, like that probably people aren't really aware about. Like, for example, bullfrog tadpoles were, back in the day, Phil Cohen discovered the urea cycle, through studying bullfrog tadpoles. The urea cycle actually is not functional, not even expressed. There's 6 enzymes in the urea cycle, none of which are expressed in the bullfrog tadpole liver. But the liver, they have a liver, it's fully differentiated and everything. But during metamorphosis, the liver reprograms itself and actually expresses permanently the urea cycle. And that is, of course, in preparation for life on land. The animals would die if they weren't able to handle nitrogenous waste. So there were fundamental. These were models back in the day for some key discoveries, and then folks discovered, xenopus in South Africa and ended up using xenopus for obvious reasons as a model system, and that kind of took off because the husbandry was a lot easier. But in adopting xenopus, for example, they're actually semi-terrestrial at best. They actually live most of their life in water. And so a lot of the key changes that happen in bullfrogs, like really going from water to life on land, don't happen as much in xenopus. So it's I guess a cautionary tale for being open to actually looking at a broad range of different organisms and not sticking with one as a model.
Yeah, yeah. With that, Caren, I think I'd like to release you from the pressure of being in the spotlight, and I'd like to take the time to thank all of the speakers. It was very diverse in the topics that they covered. I was thrilled by the depth, and it's nice to see the progression of younger people getting into the science, and then people that have like Elise who have started their careers, and then to see what somebody like yourself, Caren, has accomplished over so many years. And so on behalf of the awards committee, which I'm just one member of, Bernard, who's been on the call here, has been another member of the group and a long-standing member. We get thrilled by seeing the strengths of the scientists that are in the Canadian ecosystem that are involved with looking at this topic of endocrine disruption and how pervasive it is in our lives. That's maybe a sad commentary that it's still as pervasive as it is, but at least we know we've got strong science that's backing a better understanding of that. With that, I'd like to pass over to Romy for just her final comments and thank everybody on behalf of the Society for participating in this webinar. Thank you.
Thank you so much, Glenn, for hosting this webinar and for our wonderful speakers. Before we close, I would like to officially launch our ICEDA 26 ICEDA Awards competition. The ICEDA Awards recognize the contribution of our members to advancing knowledge and engagement in endocrine disruptor research, including scientific research, methodologies, policy, and knowledge mobilization. In 2026, ICEDA will once again offer 6 awards covering all career stages: Career Award, Mid-Career Award, Early Career Award, and 3 ICEDA Awards for trainees as the postdoctoral, PhD, and master's level. And as of today, the ICEDA Awards 2026 are officially open. Full details and eligibility criteria, application guidelines are available on our website on the ICEDA Awards page. And the application deadline is in 4 months, so you have plenty of time, but spread the word. It's August 28, 2026. And the application will be reviewed by the jury and the awards committee. And we strongly encourage our members and especially trainees to apply or to nominate a colleague. So with that in mind, I would like to sincerely thank all of the speakers today: Glenn, the Committee of Research Initiative and Excellence, and also the Evertea Foundation that funds the awardees for the trainees. And thank you for joining us today. We will record this meeting and we will upload it on our YouTube channel. And yeah, have a great rest of the day.